Welcome to the third online Brain Circle. My name is Shai Berman. I'm a PhD student at the Edmund and Lily Safra Center for Brain Sciences, or ELSEC. I'm excited to serve as this Brain Circle's moderator that will focus on the aging brain, from disease genetics to mini brains in a dish. I know that Idan's shoes are big shoes to fill, but luckily I'm joined here by two great speakers and even better scientists, Dr. Nomi Habib and Professor Eran Meshorer. Both Nomi and Eran obtained their PhDs at the Hebrew University of Jerusalem. Nomi then worked as a postdoctoral fellow at the Broad Institute of MIT and Harvard, during which she developed new technologies for genome editing and for molecular profiling of the brain at the single cell level. Currently, Nomi is an assistant professor in ELSEC, where she uses her expertise to study aging-related pathologies in the brain, combining the fields of neuroscience, computational biology, molecular biology, and genomics. Eran is the Arthur Gutterman Family Chair for Stem Cell Research and a full professor for molecular neuroscience. Eran performed his postdoctoral studies at the National Cancer Institute at the NIH, where he studied epigenetic regulation of embryonic stem cells. He's a member of ELSEC and the current head of department of the genetics at the Hebrew University. His group combines state-of-the-art live imaging with genomic approaches and computational biology to study stem cell differentiation, epigenetics, and neurodegenerative diseases. Eran and Nomi are adventurous not only in the lab, but also outside of the lab. Nomi enjoys capoeira and hiking, and I urge you all to look for the YouTube video of Eran cycling throughout Europe with his friends. Today we will hear how Nomi characterizes the single cells in the human brain and how she uses it to better understand Alzheimer's disease. Eran will then teach us how we can use genetic tools to build mini brains in the lab and how this approach can help us gain some insight into brain diseases. Today's talks are fascinating for me personally. I study the structure and function of the long connections of the brain. And like our speakers today, I focus on the human brain, both in healthy subjects and patients suffering from disease, specifically multiple sclerosis. But I use MRI to look at the large-scale structure of the brain. And today, I'm looking forward to hearing more about the advanced molecular and genetic tools that are being used that allow us to zoom into the brain. Nomi will start our event. She will be the first speaker. After she will finish, we will take a short break for a couple of questions. Then Eran will speak, and after he's done, we'll have a longer time for discussion and for more questions from you guys. So feel free to use the chat option in Zoom to write questions during the talks. I will convey them to our speakers afterwards. So thank you again all for coming, and let Nomi take it away.
Thank you, Shai, for this great introduction. So I'm happy to share with you today our research on the aging brain. And we'll start by saying that aging is actually a natural process that occurs throughout our body. And it's also a slow process. We can see signs of it on our skin and hair. And also in the brain, aging is a slow process that occurs throughout many years, but it's much less visible. So it's harder for us also to notice it and research it. So as we mature, we gain information, knowledge, wisdom, memories, and experiences. But unfortunately, advanced age also is a big risk factor for multiple diseases. Many of them are neurodegenerative diseases that are very devastating, such as Parkinson's, Alzheimer's, and Huntington disease. And in fact, we're living in an aging world, meaning that the proportion of aging individuals in the population keeps increasing, and it's projected to even become much larger over the next 30 years. So partially, this demographics is because of the advancement of modern medicine that now is become very efficient in prolonging of our life. But unfortunately, they're not as efficient in maintaining healthy aging. So we see an increase in aging-related brain diseases, many of which have no effective treatments or prevention strategies. So a very natural question for us is, how can we maintain a healthy brain with advanced age? And there are great examples showing us that it's definitely possible. But in order for us to make sure it actually does happen for all of us, we need to understand the basics of aging and neurodegenerative diseases in the brain. And many of these diseases are actually very complex. They include multiple different risk factors and very complex molecular mechanisms. So the risk factors are diverse. They include genetic risk factors, meaning the risk we inherit from our parents. And some diseases have a very strong genetic risk factor, such as Huntington disease. Others might be more influenced by the environment or even by our lifestyle, such as Alzheimer's disease. But among all of these diseases, we see a common theme, which is advanced age is the most prominent risk factor. So we're really fascinated about what happens to our brain as we age, what happens to the healthy brain, and what happens in the disease process. And we have different methods to study the brain. We could look at the high level and understand the brain anatomy and the activity of the brain using methods such as MRI scanning, like Shai is actually using in her research. But today we'll zoom into the brain and try to look at the brain at the deepest level possible, looking at cells and at biomolecules. So our strategy to understand disease mechanism is when we're trying to track and profile the brain throughout the process of aging and throughout the progression of disease. And at each of these levels, we want to find the molecules, the key triggers that are driving the process of disease and aging. So these such molecular triggers are actually the ones that we eventually develop drugs against that will hopefully help us either prevent or cure diseases. So the main question we have to ask is, how do we profile the brain on a molecular level? So in fact, we have a great arsenal of methods that enable us to profile the brain. We can take a brain tissue and extract from it many different biomolecules. It could be RNA, DNA, proteins, metabolites. And one of my favorite molecule is actually RNA, which is the one we'll focus on today because it's relatively easy to measure, and also stores so much information about the cell and its function. But the challenge when we're using molecular measurements in the brain is because the brain is a very complex tissue, unlike others. So what happens when we take a piece of tissue and try to measure molecules in it? What we're actually doing is similar to a fruit shake. We're taking many different fruits to make a fruit shake. We take many different fruits and mash them all up together. So imagine that we have a rotten fruit in there. What will happen is we might feel a funny taste from this rotten fruit, but we wouldn't know which fruit is the cause of this. Is it the kiwi or the mango? So similarly in the brain, we don't have a single cell type. It's not homogeneous, but we have multiple different cell types all combined together. We have neuronal cells, nerve cells that are surrounded by oligodendrocytes, astrocytes, microglia, vascular cells. So these are all very different types of cells. You can see how different they are in their shape, but they're also very different in their function. So when we measure molecular measurements in a whole piece of tissue, what we're doing is we then cannot separate which of these cell types is actually the driver of disease. Where is the molecular trigger actually coming from? And this is a very critical information we want to obtain. So what I've been developing over the past several years are methods to profile the brain at the much more detailed cellular level. So we're going backwards here, just one second. So what we actually want to do is we want to measure these molecules not at the tissue level, but rather at the single cell level. We want to take the brain, separate cells from it, and for each of these cells, we want to measure, get a very detailed profiles of its molecular profiling. Much more similar to these fruit salad that you can see here, where we can really go and examine each fruit on its own and understand the source of the rotten fruit. So this is exactly the method I've been developing, which is called single-cell RNA sequencing, where we are able to measure thousands of RNA molecules in individual cells. And we're not doing it for one cell or two cells. We're doing it for hundreds of thousands of cells. So we naturally can't just do it by hand one by one. We're actually using microfluidic devices which capture individual cells in small droplets of oil and water. So this technology now enables us to actually execute our strategy. We can profile the brain across stages of disease and aging by building very detailed cellular maps of the brain. For every stage of disease, we can track each of these different cell types and understand how they're changing on a very deep molecular level. So one missing piece in this puzzle is how do we actually transfer from taking a piece of brain, and here you can see a small piece of human brain that we took a picture of in my lab. How do we transfer it through our microfluidic device and end up with a very detailed cellular map? This actually requires the help of big data analysis, machine learning, and artificial intelligence algorithms in order to take such large-scale data of thousands of RNA molecules and turn it into detailed maps that enable us to track disease. So what I want to tell you now is how we're using such technology in my lab to study disease. So the disease we're actually focusing most on in my lab is Alzheimer's disease. So as you might know, Alzheimer's disease is a type of dementia, and it's actually the most common form of dementia. And it's highly prevalent. I think I don't even need to tell you how prevalent it is, because we each know people that are affected by this disease, and it's prevalent around the world. So at the clinical stage of the disease, Alzheimer's affects our speech, our mood, our memory, and it leads to cognitive decline. But at the last stage of this disease, it also leads to really ruining basic bodily functions and leads to death. But what happens at the molecular level? What we know is Alzheimer's disease is a progressive neurodegenerative disease. What does that mean? It means that there are molecular damage that accumulates in the brain, and this we know happens probably over at least 20 years, that there is molecular damage that accumulates. And what does that cause? It causes that nerve cells, these cells that are so crucial for the function of our brain, are being damaged and die. So one of these molecular damages that we know of is accumulating of proteins, of different proteins. You might have heard of amyloid beta plaques, for example, that are getting accumulated in our brain, and one of the causes for these neuronal damage. But in fact, for over 30 years, we've been studying Alzheimer's disease, focusing on these two aspects: the damage that is caused to neurons and the accumulation, the protein aggregates such as amyloid plaques. And in fact, we still have no effective treatments or prevention strategies. So what that caused us was a call for us to think outside of the box and how can we study Alzheimer's from a different perspective. So we decided to do that by actually broadening our view. So in fact, as I explained to you before, the brain doesn't look like the picture you can see here on the left of neurons that are being alone in the brain, but rather there's a very complex cellular environment of the brain where neurons that here in the cartoon are in blue are surrounded by multiple different cell types. And we know today that many different cells beyond neurons are implicated in disease progression. So what we asked ourselves is, can we take a much more unbiased view and try to find new cellular drivers of Alzheimer's disease? So we first actually turned to mouse models, which is a very good way to start because it's such a clean system. We have a mouse that is healthy, and we have a mouse that almost is exactly the same to that other mouse, but it's a model for Alzheimer's disease, and at certain age starts declining cognitively. And this is a great collaboration driven by several students in my lab with many collaborators around the world. So what we did is we took the wild type the healthy and the Alzheimer's disease mice, and we use the single nuclear RNA sequencing technology that we have in order to profile their brain along different stages of the disease. So we measured thousands of RNA molecules in single cells, and we measured more than 200,000 cells. And we used that to build a very detailed map of the Alzheimer's brain. So I can tell you already, I'll jump to the conclusion of this research, that what we found to our surprise when we looked unbiasedly and asked which of all of these cell types are changing the most dramatically in Alzheimer's disease, what we found was that the cells that were changing most dramatically were actually astrocyte cells. So what are astrocyte cells? Astrocytes are star-like shaped cells that appear in the brain. You can see here an image of them here on the left, taken in a microscope. And they have critical role in the brain in maintaining a healthy and normal function for the brain. It's actually the cell that has the most functions I've ever actually researched. It takes a role in brain metabolism, maintaining the homeostasis in the brain, in the integrity of the barrier of the brain, in maintaining neuronal function, and they also play a critical role upon injury of the brain. And so there are many, many different functions that they are holding, but they weren't as known to be involved in Alzheimer's disease. So let me tell you what we found. We found in a nutshell that astrocytes are not all homogeneous. They're not the same throughout the brain, but rather we can find 3 very different populations of astrocyte cells. So they're different in their shape and they're different in their function. But what happens in the healthy brain, we mainly have this population of astrocytes we call homeostatic astrocytes. What happened in the Alzheimer's brain is it was a complete switch from the homeostatic state to a completely new population of astrocytes that we call disease-associated astrocytes. So these disease-associated astrocytes, we could actually image them in the brain and see that they are positioned very closely to these protein aggregates called protein plaques. We actually also noticed that they appear very early during disease progression, before we see any signs of cognitive decline, really suggesting they play an active role in the disease progression. And finally, we also find them even in natural aging. So that's really intriguing. So overall, we think we found a really critical new player in Alzheimer's disease that might be even good as a drug target. But we really wanted to understand how is this relevant to the human brain. So mouse and human brains are inherently different from each other. But actually, the single-cell RNA sequencing technology we developed enables us now, for the first time, to also profile the human brain in such detailed view, because it's applicable for frozen tissue. So we could take post-mortem human tissue of donors throughout the world that can get shipped to our lab so we can profile them at the molecular level. So what we did is we started this great collaboration that was led by Anael Kain, my student, with many different collaborators, mainly Phil DeJager from Columbia University, where we profiled both healthy individuals and individuals that have Alzheimer's. And here you can see how our data actually looks. I'll train you as a neuroscientist. This colorful image that you're seeing here is the map of the brain that our algorithm is generating, where every little cloud of color actually captured a different type of brain cell, a type of cell in the brain. So we can now look at each of these different brain cells separately and ask how they're changing between healthy and diseased individuals. So first, just one word about our maps. So because our maps are so detailed, we're actually very excited about them, because we now can actually discover many more things we weren't even asking. We can find new diversity of cells, new cell populations in the brain. We can see how the brain cells are changing across different individuals. Here we profile 24 different individuals. We can look at differences with age and disease, and also we can see differences, very striking differences between the women and men, where these differences are not contained in a single cell type, but rather every cell different cell has a difference between the two sexes. So let's go back to our main question, which was how does the brain change with Alzheimer's disease? And for that, we're actually comparing directly the Alzheimer's individuals with the healthy individuals, and we really are measuring changes throughout different cells. But for today, we'll focus on what happens with astrocyte cells. So here you can see how we zoomed into astrocyte cells. So again, you're seeing a map of our data where every different color represents a completely different population of astrocytes. So what you can really see here clearly is we found at least 5 different populations of astrocytes, just showing how complex is the human brain in compared to the mouse brain. But if we really compare the human and mouse, we see that actually there's something very similar here. Most individuals have a very large population of these homeostatic astrocytes, and then different individuals have different population of reactive astrocytes. And we asked ourselves, can one of these populations be actually associated specifically with Alzheimer's disease? And what we found is indeed this population marked here in blue, this reactive state of astrocytes dramatically increase in the Alzheimer's brain. And we find this not only in these 24 individuals, but in a much larger cohort of brains of 640 individuals. So again, we're now placing astrocyte cells as potential drivers of Alzheimer's disease, which gives us a lot of hope because we have new avenue to actually look and research. But just before I end, I want to kind of remind you that when I started my introduction, I said that the brain is very complex and there are many different cell types all kind of together in the brain. And these cells are communicating with each other. So you can really understand that if we're going to try and find a therapeutic avenue that only targets a single cell, it probably won't be complete because we can correct a single cell, but its environment might still be toxic and inflammatory. So what we really want to understand is how all the cells are changing. And indeed, when we take our maps, we can not only look at a single cell type, but we can look across all the different cell types, and we see that all of these cells are dramatically changing when we look across individuals. So we finally asked ourselves, are these changes in each of these cell types, is it actually happening independently in each of these cells? Or can we find something that is coordinated across all of these cells? And can we find a connection to Alzheimer's disease? And indeed, when we take all this data together and we combine it and we separate our individuals to cognitively healthy and cognitively declined individuals, we see a specific cellular environment that appears to be very, very specific to the Alzheimer's brain, where we see changes in all the different cell types, in neurons, oligodendrocytes, astrocytes, endothelial cells, and so on. And this cellular environment is not only found in the 24 individuals we profiled in the single cell level, but we also were able to then validate these findings in a much larger cohort of 640 individuals. So we're very excited about this opportunity to really characterize a new cellular environment that's specific to the Alzheimer's brain. So I'll summarize and say that what I showed you today, I started by showing you a new technology that we have, a new tool that we have in our hands called single-cell RNA sequencing, which enabled us to look at the brain at much more detailed resolution, which we were ever able before. So we use this technology to build detailed cellular map of the Alzheimer's brain in mouse models and in humans. And we use these maps to identify astrocyte cells as potential new players in Alzheimer's disease that might be very critical. And we then take a much larger view now and combine this all together in finding new unique cellular environments in the brain. So this is all really exciting, but there also remains many unanswered questions. So mainly, I presented here a hypothesis that astrocyte cells in this unique cellular environment we're encoding is driving Alzheimer's disease and potentially could be a great drug target. But what we're left to do is actually prove this, test our hypothesis. And to do that, what we need to do is go back and take a live human brain and manipulate it, actually change cells and genes in this brain. So this seems to be impossible since we are not going to approach an individual and actually change its brain. What we actually need to have is have a small brain in a dish that we can play and manipulate. So this seems science fictional, but it's in fact what Eran is going to tell you about how he is doing that in his lab today. So I'll just end by saying a really big thank you for everyone involved. So many great people in my lab, and it's really great fun to work with them. My collaborators, different funding, and the brain donors, emphasizing that science is a team effort. So we need all of the people together in order to make it work. So thank you.
Thank you so much, Naomi. It was really fascinating. So before we move on to Eran, let's take a couple of questions. I see that Idan asked, I think, the question that we all want to know, which is, you actually showed us that the astrocytes and other cells are different in Alzheimer's disease. And the question is how one can correct a sick cell. Can we do anything to repair cells?
That's a great question. So we have different ways to repair cells. Actually, Eran is going to discuss briefly some of these more genetic manipulations and how we can manipulate cells on a genetic level using, for example, the CRISPR technology that I was fortunate to be part of its development during my postdoctorate training. But there are also other ways. So if we understand the function that is changing, we don't need to change the cell completely, we just need to change a single function. And that could even be happening by using the right small molecule that would actually signal the cell to change. So we're using the same pathways that the cell is using to communicate. We can also use that as a driver to change the brain.
That's really cool. I actually also have a question myself. I was wondering, you showed differences between healthy subjects and sick individuals after a postmortem. I wonder if there's a lot of variability between the diseased brains. Is there more variability in diseased brains more than in the healthy brains? And if this is an advantage, meaning that maybe we can use that later on for personalized medicine, or is it troublesome because it hurts the way we can treat them?
Yeah, this is a really great question because it actually highlights one of the challenges of studying the human brain. So people are very diverse. We're diverse in our genetics, we're diverse in our lifestyle, and this is reflected in the brain and in the measurements that we're doing in the brain. So what that means is that when we're looking at disease, we're adding another variability, which is the type of disease you have. So Alzheimer's is a complex disease, and we definitely know today that we can subcategorize it. We just don't know exactly what is the best way to do that. And we definitely see that in the brain. We see that a sick individual is not always identical to a different individual that is sick. So actually what we need to do is increase the number of people that we're doing, and we are working towards that in our lab. We're actually increasing our sample sizes from 24 to 500 brains. So hopefully we'll have a much better way to address this diversity.
Okay, thank you so much. Before we move on to Eran, I'd like to remind you that you can still raise questions in the chat, even for Naomi, but also for Eran during his talk, and I will later convey them to our speakers. So thank you again, and Eran, take it away.
Oops.
Great. All right. That was a beautiful talk, and what Naomi described to you so far is how she takes brains and basically deconstructs them into single cells. And basically, she's analyzing these single cells to understand the whole brain. What we're trying to do is almost the opposite direction. We're trying to start from single cells, stem cells, basically. I'll show you what they are in a minute, and trying to rebuild mini brains in a dish. So it's kind of the opposite, but really complementary approaches to the same problem. So in our lab, we're really trying to model neurodegenerative diseases, such as actually one of the diseases that Naomi mentioned, Huntington's, which is a genetic disease. We're trying to model them in the dish. So where are my slides? So I'll just begin by saying that my
Yeah, you can transfer one. Maybe it will start.
Oh, okay, good. Here we go. Yeah, so I'll just start by telling you what embryonic stem cells are and how they can help us. So let's go back to our very first week of life. What you see here is just after fertilization. Okay, this is basically the human egg immediately after the minute of fertilization. And what is this egg is going to do is going to divide into two cells. It takes about a day, and then each of these cells is going to divide again. And in the next few seconds, you'll see the entire whole week of human development. So here you can see immediately the first division, and then each of these two cells will divide again. So you have a 4-cell embryo, and then each of these 4 cells divides, and you have 8 cells, and 16 cells, and 32 cells, and so on and so forth, until you get this pack of cells at about day 4, 5, and 6 during development. But at about the 7th day, something remarkable happens around here. Okay? Suddenly you see 2 cell populations. One is this inner cell mass. It's this ball of cells that is inside this bigger hollow ball. And these very cells Okay, so these very cells that you can see here are going to develop into the entire embryo. And the outer cells, all this bigger ball, is going to develop into the placenta. But these cells have the ability to generate each and every cell in our body. So this is really remarkable, and about 2 months later, this pack of cells, the inner cell mass, will look something like this. So this is a 2-month-old embryo. And these embryonic stem cells, when we actually grow them in the lab, they have 2 remarkable characteristics. One is that embryonic stem cells can self-renew indefinitely. It means that, just like I showed you earlier with the egg, each cell can form 2 identical cells, and so on and so forth. So I can actually take one cell, a single cell, and in fact fill the entire room with identical cells. And this is very important because if we are to use embryonic stem cells for future medicine, for example, to try and generate livers or other tissues, we'll need massive cells. It's not enough to take a few of the cells from the embryos. It doesn't matter how many embryos we're going to collect, we'll need really tons and masses of cells. So this is a very important characteristic that it happened to be for embryonic stem cells growing in dishes. But the other, maybe just as important characteristic is that embryonic stem cells also are what we call pluripotent. Potential, pluri a lot. So they can basically form all the cell types of the organism in a dish. So here in this cartoon, you can see these embryonic stem cells, right? So that when we grow them in a dish, this is the embryo. And if we give them the correct conditions, and these conditions are being worked out in many different labs all over the world, we can transform them into different cell types, such as fat cells, blood cells, and different brain cells, such as neurons or even astrocytes that Nomi talked about in her talk. In this movie, you can see embryonic stem cells that have been developed into heart cells. And these cells start to beat spontaneously. Just by giving them the right proteins and the right conditions, they form heart structures which have an inner pacemaker, and they just start beating spontaneously. This is always remarkable to see small beating hearts in a dish in a lab. In another example, you can see here embryonic stem cells differentiating into neurons. Here you can see these colonies of embryonic stem cells. Always in the upper left corner, you'll see one cell. This is an individual embryonic stem cell. This is a stem cell colony. And this is of about 4 or 5 days in differentiation. You can see they form these so-called embryoid bodies. They're hollow balls of cells. And then we can replate them in specific media, in specific conditions, and cytokines and proteins and all sorts of goodies, that they start forming neurons. So they generate these nice neurons, connections, and here after about a month they can generate a nice, relatively homogeneous neuronal network. But there are issues regarding use and growth of embryonic stem cells. So the first one is rejection, because if we are to use embryonic stem cells, for example, for transplantation in humans, of course they were derived by an in vitro fertilized embryo, and if we're going to transplant them, we're going to have all the issues of rejection, just like tissue transplantation, liver, kidney, etc. The other problem that might arise is that they might form tumors. Remember, I told you that embryonic stem cells can self-renew indefinitely. From one cell, I can fill the room with cells. So what happens if we are going to generate a liver and then transplant this liver in humans? Are we going to have maybe some cells that have not been differentiated and might form these tumors? So this is a safety issue that we have to take into consideration. And another issue is there is an ethical issue regarding embryonic stem cells, because of course we need an embryo, a fertilized human embryo, in order to generate them, and in some societies this is less acceptable, in others it's more acceptable. Just a note, in Judaism, life begins at day 41, whereas in Christianity, life begins immediately after fertilization. And this is why there is this tension regarding the ethical issue in Christianity. Okay. So are there alternatives? The answer is yes. So let me introduce you to Shinya Yamanaka and John Gurdon, two people separated by many years, both of whom actually showed that at the cellular level, you can reprogram mature cells into embryonic-like cells. Okay, so John Gurdon actually did it by cloning. He cloned frogs in the 1950s and early 60s and showed that at the single cell level, if you take a mature cell and you introduce it into a fertilized egg, this egg can develop into a whole organism. Many years later, Dolly, the first mammalian clone, was born. And Shinya Yamanaka showed that you can directly reprogram cells into embryonic-like cells. They both shared the Nobel Prize in 2012, just 8 years ago. Actually, it took Shinya Yamanaka 5 years to reach this summit. And John Gurdon, as I told you, his experiments were in the late 50s and early 60s, so somewhat longer way to reach it. Okay, so let's go to Yamanaka's experiment. What Shinya Yamanaka showed us, that you can actually take mature cells, what we call somatic cells, like skin cells, any type of cells, and you can actually introduce stem cell factors. These are proteins that are expressed at high levels in embryonic stem cells. They are depicted here by these 4 colors. you can introduce them into the cells, into these skin cells, and within a matter of 2 or 3 weeks, these cells will gradually reprogram into so-called induced pluripotent stem cells. Okay, they are induced because we reprogram them and they are pluripotent. And in fact, when he published the first paper in 2006, everyone was very skeptical, can these cells really be as good as embryonic stem cells? He did it first in mouse, and it took a year or two until this feat was also accomplished in humans. But in fact, you can generate mice that are healthy and fertile from these induced pluripotent stem cells. So it means that these are really, truly pluripotent, and they can generate whole organisms. So we believe that the human counterpart, of course we can't generate humans, but we believe that the human counterpart, the human induced pluripotent stem cells, are as pluripotent as the mouse cells. So now we can go to patients like Huntington's disease patients, or Alzheimer's patients, or Parkinson's patients, and just take a simple skin biopsy, or even blood, or even just the cells from the hair, and reprogram them into these induced pluripotent stem cells, into iPS cells. Now, I already told you that we can differentiate these stem cells into different cell types. So this allows us to generate specific cells that are genetically matched to the donor. Okay, so this is really remarkable from the potential of this technology. Okay, so in Huntington's disease, it's a neurodegenerative disease. As Nomi explained, these are a family of diseases in which cells, brain cells, are dying. In Huntington's disease, there is what we call striatal atrophy. Cells specifically in the striatum die. These are the specific cell type, GABAergic neurons, medium spiny neurons, doesn't matter, but you can see here the region of the striatum. And here you can see this striatal atrophy. You can see that there is larger holes, larger cavities in the Huntington's brain as compared to a healthy brain. So I'll take a minute and introduce you to some molecular biology, just so we'll understand the genetics of the disease. As you may know, DNA is comprised of 4 letters, 4 bases. We call them A, G, T, and C. And this DNA basically encodes all the proteins that we produce inside the cells. Now, in Huntington's, there is one gene in our body that is called huntingtin, because when it was discovered as the gene causing Huntington's disease. You can see here the sequence of this gene. You can read this different letter, but suddenly at a certain point, you can see this CAG, 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 CAG. It goes on for like 20 CAGs in a row. This is in healthy individuals, okay? This is how our genes look like. But in Huntington's disease, instead of 15 or 20 of these CAGs, you suddenly get over 50, over 40, over 50, over 60, sometimes more than 100 of these CAGs. Now, what does it do? CAG encodes a specific amino acid. So when the DNA is encoding proteins through RNA, which Nomi talked about, it encodes amino acids, which are the building blocks of proteins. CAG encodes a specific amino acid called glutamine, which is denoted by the letter Q. Okay, so all we need to know so far is that inside this huntingtin protein, we have a region of Q, 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 one after the other, which is encoded, remember, by CAG, 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 etc. Okay, so this is the healthy huntingtin. You see there's anywhere between 15 and 35, 39 CAG repeats, and this would be the Huntington's disease CAG, and here you can see the protein. So here we'd have a short polyglutamine or polyQ protein, and here we would have a long polyQ protein. So these diseases actually are called polyQ diseases because of this type of mutation. Okay, so what does this polyQ does? In Huntington's disease, the protein has issues with folding, and therefore you get this massive aggregation of the polyQ protein. So this is just a cartoon to show this aggregation problems in the diseased neurons. And here you can actually see cells. Here you can see the healthy Huntingtin with 25 Qs, right? This is the healthy range. But here you can see 94 Qs. And you can suddenly see that instead of this nice diffuse green color in the healthy neurons, suddenly you can see these aggregates. You see these very round, sharp aggregate formations of these green fluorescence. So this is what happens in the neurons, and after a while they can't cope with these aggregates anymore and they die. So to model a disease, we can take embryonic stem cells or induced pluripotent stem cells, either or, from embryos or directly from patients. And we can now use the CRISPR tool. This is a genetic tool that allow us to edit DNA, to change bases inside the sequence, and basically correct the DNA from a disease state to a healthy state. So this is what we can do. This is what we're doing in the lab. We're taking cells from patients, we're reprogramming them to iPS cells. So these are now disease-carrying iPS cells, and we correct them using CRISPR technology to a healthy DNA, to a healthy cell. Okay. And here you can see, for example, neurons, mature neurons that are the specific neurons that are dying in Huntington's disease. And this took about several years just to generate this type of neuron, but we can actually do this now in the lab. But we can not only generate neurons, we can also generate these so-called mini-brains. So using specific technologies of three-dimensional differentiation instead of two-dimensional differentiation, we can actually generate these cerebral organoids, or in short and simple, mini-brains. So instead of just differentiating the cell in a two-dimensional plastic plate like we usually do, we basically rotate them in these little dishes, and they start forming these three-dimensional structures. So if you give the right conditions, they start look like this. So after 5 days, 7 days, 12 days, I'll show you in a minute, more mature mini-brains. But we can do it with the healthy stem cells, and we can do it with the diseased stem cells. And actually, they form really nice structures that really resemble brain structures. You can see here slices, microscopic slices through these mini-brains, and you can see the different cell types. This is different stainings, red and green stain different populations of neurons that if I label them together, you can see that they label different types of neurons. So just to end, I'll show you some beautiful images, microscopic images from our brain tissues. Again, you can see these different types of neurons stained by different markers. And again, you can see this assembly of the images together. These actually have been grown in the lab for 5.5 months. You can see a zoom in into these structures. You can see the nice neurons. Each and every one of these circles is actually a nucleus of a neuron. You can see the neuronal projections. Here again, a zoom in, different cell types. You can see the the cells together. And again, a closer look at these neurons. So the nice thing about this is that we basically generate a heterogeneous tissues, not only with neurons, but also with the different cell types of the brain, microglia, astrocytes, oligodendrocytes. So we have all this brain heterogeneity in a small tissue-like sample. So just to end, what are we doing with these neurons and with these mini brains? Now we can really screen for relevant compounds, and we can actually use the disease pathology as a readout for our screens. For example, I told you that in Huntington's disease, these defective proteins are forming these aggregates. And what you can see here are neurons, and here you can see these defective aggregates. And now we can start using drugs, drug combinations, small molecules, other proteins, to try and search for ones that alleviate these aggregates. So we're actually using computerized images just to screen these compounds very fast and very effective. And we're really starting to find proteins and molecules that affect these cellular aggregates. So again, science is, as Nomi mentioned, a team effort, and all thanks to my team and the funding and everyone else. And I'll be happy to open this for discussion with Nomi, Shai, and yourselves. Thank you very much for being here.
Thank you so much, Eran. It was truly a science fiction research. So I want to start with just something that really came to mind. You showed us how Huntington's disease is a one mutation disease, which seems unique. So I wonder if that's a criterion, if there is something you require to be able to produce in a mini brain or to study in general. Is there a specific type of family of disease that you can study, or is it more general?
Yeah, so this is a very interesting, excellent question, because the reason we started with Huntington's is exactly because of that. So we were looking for, of course, a brain-related disease, a neurodegenerative disease, but which has a very strong genetic background. And in fact, in Huntington's disease, if you carry more than 40 Qs in your protein, you have a 100% chance of developing the disease. So it's the penetrance, what we call, is full penetrance. And in fact, many of the genetic diseases, you need 2 mutations because we have 2 sets of chromosomes. In Huntington's, only one of the chromosomes usually is affected, and it's what we call a dominant disease. So one mutation in only of the one of the 2 genes, you have full penetrance, and you're going to develop the disease and probably die from the disease. So this allows a very strong tool because we know that the cells have the mutation, and we can actually correct them using these genetic tools. So for studying, for research, it's a very, very powerful approach.
Thank you.
If I can add.
Of course.
What's really, I think, I find exciting about the iPS cells is you could actually take also any sick individual that does have some genetic basis, even if it is of multiple genes, but you do have the opportunity to now generate a mini brain from this individual, even if you don't know the driving gene. Is that correct?
Absolutely, yes. So one of the interesting things, so as I said, we started with Huntington's because it was, everything is known. We know the mutation and we know that this particular cell is sick. But our goal was really to go into more complex diseases such as Parkinson's, for example, or even Alzheimer's. So there are cases where Parkinson's is purely genetic, but this explains only only the minority of the cases all in all. But we can take cells from a Parkinson's patient and really learn about the disease using these mini brains. So this is something that we're definitely interested in. Thank you.
So I have a really great question from Adam Craig. He's asking whether the aggregates are in the intracellular space and whether we can identify which cell types are producing them. I guess if we could, maybe we could combine both of your methods together.
Yeah, so this is, again, a very interesting question. The aggregates are indeed intracellular, and they actually can be found both in the cell nucleus and in the cytoplasm. And what we don't understand is why certain neurons, certain cells, have these aggregates and others don't. Because huntingtin, the protein itself, is expressed not only in the brain, but in the entire cells of our body. So in the skin cells and all the tissues, liver, kidney, blood, etc. But only certain cells in our brain have these problems with the aggregates. And these are these spiny neurons in the striatum, GABAergic cells, and these are mostly the ones that get affected in Huntington. And this is one of the riddles of Huntington's disease, which we absolutely don't understand. We don't know why, but it's a great question.
And we can actually expand this to all the different diseases and asking, I think many scientists around the world, including ourselves, are asking what makes a specific type of neuron vulnerable to a disease. And just to emphasize this point, the DNA, the genetic basis, is identical throughout all our cells, right? So if we have a mutation in one cell, we have it in the other. In addition to that, also if we're exposed to something in our environment, we would expect almost all of our cells to be exposed to that. So all of these factors together, if we're eating differently, it would affect all the cells in our body differently. And still we see, so you have this great example from Huntington's. We can see it throughout all the different neurodegenerative diseases that we always have a specific cell that's more vulnerable than others, a specific brain region that's more vulnerable than others. And this is one of the questions we don't have answers for, and it's just fascinating.
Great. So I have, inspired by Dan's question, I have actually a question for both of you. Eran, you mentioned CRISPR, and Nomi, you work with CRISPR as well. You showed that we can use it in some therapeutic ways, perhaps to grow a liver. So the first question is whether it can be used in vivo to repair diseases or perhaps specific cells that are damaged. But I was also wondering if you could kind of improve things, not from a disease standpoint, if you can make yourself taller or stronger, and maybe you would like to say something about the ethical implications of this as well.
No, you will start with I'll go into the human embryo, but you can start with in vivo CRISPR.
With the in vivo CRISPR. Yeah. Okay, so CRISPR is an amazing tool, and I'm saying it unbiasedly, really opened many opportunities to manipulate our genome, to change the code, the genetic code that we're born with in a very efficient way. And this is very exciting. If Eran can correct Huntington in a dish, we are definitely very intrigued by correcting Huntington in a live human individual or in an embryo. So while this is ongoing, there are many efforts around the world in order to do this. This is something you might hear of as gene therapy, and there's already a few clinical trials ongoing, and there's even like the first individual who just got a treatment for one of these, for a devastating disease. There's a famous story about a Chinese researcher who actually generated CRISPR babies. And here's where we start to cross the line, right? What is ethical and what is not? And this is such a strong tool that we do need very strong regulations on how to use it. So definitely we do not want to change our height or the color of our eyes, and even not our intelligence. So how do we not cross the line is something as scientists we need to work along policymakers to make sure it actually happens.
Right. So just to maybe follow up on what Nomi just said is that there is a possibility. I mean, the technology to actually modify the DNA at the early embryo just after fertilization, or even before fertilization, this technology exists. So this is exactly what He, the Chinese researcher, did. He tried to modify a particular gene. He thought that this would confer some resistance to AIDS. He probably wasn't successful. But there are two ways to look at it. One is really going into the embryo and changing, traits, which I think is really crossing the line. But the other is already at the adult stage, right? And just trying to change something specific, like a mutation in particular cells, because maybe the mutation is not affecting other cells, et cetera, et cetera. So I think that CRISPR is a very, very powerful tool that will definitely be introduced into medicine at the coming years.
Right. So I have a question for both of you about timing, slightly different. Naomi, you described how the greatest effect you see is in astrocytes, right? These are the star-supporting cells. And you also mentioned that the astrocytes have many, many roles, and they're in communication with other types of cells in the brain. So I wonder if you looked at different stages of the disease, if you would maybe find a greater effect in other types of cells. And then we can then ask.
Okay. Wow, that's a tough question.
Sorry.
And it's exactly what we're seeing, actually. So I obviously simplified the story today by focusing on astrocytes. I think I gave a glimpse of this at the end of my talk, where I said that actually what we are seeing is changes throughout multiple different cell types. And as we start kind of going one by one, looking at each of these cells, now that we have the technology to do that, we really see many different players. And one of the challenges is really putting this on a timeline, trying to see, so who's the first trigger, who is the driver of these events? And this is actually something we don't have an answer for completely. It's a little easier for us to do that when we're doing it in mouse models. When we're using human subject, which is much more informative for us, we can't do that because we can't sample the brain at multiple stages along disease. We can't sample the same individual again and again, at least with the current technology we have. So we are trying to do that by using multiple individuals that each are along different stages of the disease. So we're starting to build these models. We're starting to build models of communication. We have some initial ideas, but that's actually where we are.
Okay.
So maybe just to say something about the cellular models is that unlike the human diversity, when we are correcting a mutation, one of the big advantages is that we have what we call isogenic cells. So these are cells that have exactly the same DNA, exactly the same genetic background, only one carries the mutation and the other doesn't. So we can really compare them side by side and really ignore all the heterogeneity and all the variability that we have. So using these isogenic systems, we can really learn something, I think.
Right, so I'll replace my question with the better one from Adam Craig. He's asking, since the mutated Huntington gene is expressed in most cells but only affects a specific subset like you described, can you look at whether there are differences in post-translational modifications? And maybe before you answer, you can say what that means.
Okay, great question. So post-translational modifications are modifications that come at the protein after it's translated. So going back to our lesson 15 minutes ago, in molecular biology, DNA turns into RNA, and RNA turns into protein. So this is what we call the central dogma. The information goes from DNA to protein. Once a protein is generated, there are certain chemical modifications, like phosphorylation or even addition of sugars that stabilize the protein. And the question that Adam is asking is whether these changes might be different in a mutated protein and a healthy protein. And this is really a very good question, tough to answer. The answer is definitely yes, in short, because there are so many different modifications in the protein. Almost in all region of the protein, we can find these modifications. So in this this part of the protein that we have the mutation, there are several modifications that must be different. But at the same time, it doesn't mean that this is what is causing the aggregation, for example. Because if we are to take this polyQ, even without the huntingtin, just the polyQ, it's going to form an aggregate. So we're pretty sure that this is the main issue, the main problem. So there are many layers, many different layers to the disease, which I'm really don't have a lot of time to go into, but this is just maybe one of them, and it's really I think interesting to think about it. So thank you for the question, and we can
you'll follow up.
Yeah.
Okay, so I think one last question is just to, before we finish with Eyes to the Future, maybe you can say with just 2 sentences, where do you think your field will be in 5 years? Either of you can start.
I'm actually, maybe not surprising, very optimistic. I think that the main thing is what I see is that similar to what we are doing in the study of aging and Alzheimer's and neurodegeneration, we're looking in a very different way to how people have been studying the disease over the last 30 years, and we have much more powerful technology in our hand. So I really believe we'll have a completely different view of the disease in 5 years, and hopefully also some therapeutic targets, and also just strategies, helpful prevention strategies. And I think that's actually where we'll see the big change.
And from the stem cell point of view, so the big promise of stem cells is that they would be able to really correct some tissues. For example, I guess the lowest hanging fruit that people thought of in this regard is beta cells from the pancreas, because diabetic people, they don't have these cells are dying. So if you can transplant embryonic stem cells that have been differentiated into beta cells, you can actually correct these diseases. The same goes for Parkinson's. About 2 months ago, the first paper was published with a human being that was transplanted with iPS cells that were, induced pluripotent stem cells that were differentiated into neurons, the specific type of neurons that are dying in Parkinson's disease. These are dopaminergic neurons, and this patient was transplanted with these cells. And there's now 30 patients enrolled in this study, and many of them were already transplanted. And really the great promises in this field is that the stem cell will be able to replenish tissues. Hopefully it will be able to replenish brain tissue as well, but maybe liver transplantation and other tissues will benefit from stem cells in the not so far future, I hope, between 5 and 10 years away.
Okay, thank you so much, both of you. Thank you everyone for joining. I think you can all unmute your microphones, or maybe before you unmute yourselves, I will say that the next Brain Circle will be sometimes during mid-October. It will be for kids between ages 10 and 18, and it will be an active experience in Zoom rooms. So that sounds very cool, and I hope I'll be allowed to join even though I'm over 18 years old. So yeah, before we say goodbye, please try if you can to open your cameras and open your microphones so we can all say thank you and goodbye and hug each other.
Thank you so much.
Hello, thank you. Thank you, Francois, Emily, Lucille. See you all in the next one.
Good.